What's up, guys? Study times here. In this video, I'm going to talk about the ways that the cells can die. And if you like the video, please like and subscribe to our channel and turn on the notification bell so you won't miss any of our uploads. Let's dig into it. So, the cells have two ways in our body to die. One of them is apoptosis, and the other one is necrosis. These are different, and the way the cells do it is different too. So in apoptosis, it's a programmed cell death. So the cells are programmed and planned to die. But in necrosis, the cells just die because of some diseases or some injuries, like very high temperature or very acidic or basic environment or low amount of oxygen or nutrition. They just die because they cannot survive, basically. But in apoptosis, they are planned and programmed to die. One of the good examples is skin cells. So the old skin cells are dying and they are programmed to die, and they are getting replaced by the new cells, the young cells. Another example is fetal hands and feet. So when the human being is a fetus, the hands are actually the fingers and the toes are attached to each other. But the cells between, like here, between these fingers, between the digits, they start to die from apoptosis. They undergo apoptosis, and they make different digits, separate digits. Okay, about necrosis, one of the examples in when the blood vessel gets clogged, like to your big toe or some other blood vessels to other part of the body, you have ischemia. It means the amount of blood that going to that part is going less, so that part doesn't have enough oxygen and nutrients, and that makes the cells to die because they need oxygen and nutrient to survive, so that makes them to die. This is an example of necrosis. See, it's about some diseases or some injuries. It's not programmed. They wanted to stay alive, but they couldn't. About apoptosis, let's dig into it. So we have two pathways for apoptosis. One of them is intrinsic and the other one is extrinsic. Intrinsic, another name for it, is mitochondrial pathway because the mitochondria is doing the main job. But extrinsic pathway is death. Another name is death receptor pathway. Is extrinsic because the stimulus is from the outside, and this is intrinsic pathway because the stimulus is from the inside. So we're going to talk about each. The first one is intrinsic pathway. It can happen due to hypoxia, which means the lower amount of oxygen, or radiation, or oxidative stress, or when the amount of calcium ions in the cells are high. You can have the intrinsic pathway. These are the stimuli actually for intrinsic pathway. So how that happens? This is my mitochondria in the cell, and this is my cytosol. Like here is around it is the cytosol. We have two proteins, BAX and BAC, in the cytosol. And due to these stimuli, these BACs and BAC can go and pierce the outer mitochondrial membrane. So they go to the mitochondria and they pierce the outer mitochondrial membrane, as you can see here, and they can make it porous and leaky. So it's so leaky now at this point. Two other proteins in the between these inner and outer mitochondrial membrane are SMAX and cytochrome C. Because it becomes the outer mitochondrial membrane becomes porous and leaky, so these two proteins can go out to the cytosol. And we're going to talk about each separately, that what they do inside the cytosol. At this point, we have the SMAX and cytochrome C in the cytosol. These MACs bind to some proteins that they usually inhibit an apoptosis. So they bind to apoptosis inhibitors, and they inhibit apoptosis inhibitors basically. So it actually stimulates the cell to undergo apoptosis. And cytochrome C binds to ATP. So these ATPs are the cell's energy. So it binds to ATPs and it doesn't allow ATPs to work normally. So it actually doesn't give the cell the enough amount of energy. Another thing that cytochrome C does is it binds to APAF1. APAF1 is a kind of enzyme, and these two together make some structure called apoptosome. This apoptosome undergoes and activates procaspase 9. These are some proteins in the cytosol. And this procaspase 9, it when binds to apoptosome, becomes activated version, and it becomes caspase 9. And caspase 9 activates caspase 3. Caspase 3 activates other caspases. And these caspases, what they do, these are enzymes, and they can cleave the proteins of the nucleus or organelles or cytoskeleton. So they are proteases basically, and that makes the cell to undergo apoptosis. When the caspases destroy the cytoskeleton and proteins that connect the cytoskeleton to the membrane, they develop some blebs. We call it blebs, like as you can see in the picture, like these are blebs or some bulges, because the cytoskeleton is getting weak. So these blebs are structurally so weak, so they start to breaking off from the cell membrane. And as a result, we're gonna have macrophages that they go and engulf these break off blebs, and they phagocytose them and they destroy them. So they clean up the body from these blebs. Another way for the apoptosis, as we saw here, one of them was intrinsic pathway and the other one is extrinsic pathway. So about the extrinsic pathway, the signal actually comes from the outside. How it works? So when a macrophage just sees a cell, an old cell or pathogenic cell, a cell that has a problem. So macrophages release a type of substance called tumor necrosis factor alpha, or TNF alpha for short. And the cells have a receptor called a death receptor. This tumor necrosis factor alpha binds to the death receptor and makes the cell to die. One example is here. We have the death receptor here, and the TNF alpha binds to that death receptor called TNF alpha receptor 1. And this receptor has a domain inside the cell, and it's the death domain. When the TNF alpha binds to that receptor, this domain changes its shape, and that activates two proteins in the cytosol, FAD and TRAD. And these proteins bind to each other and make another protein called disc. How I remember these three names? Just remember tough disc. So FAD, TRAD, and they make disc. And this goes and activates procaspase 8, and procaspase 8 becomes caspase 8, like the activated version. Caspase 8 activates caspase 3, and caspase 3 activates other caspases, and they make the cell to undergo apoptosis again, like before. Another example is when a cell is expressing a foreign antigen on its surface. When the cytotoxic T cells recognize these foreign antigens, they and make a protein called Fas ligand, and they express it on the membrane. The Fas ligand goes and binds to Fas receptor, which is another type of the death receptor. And because it's a death receptor, so it has a death domain inside the cell. And when this Fas ligand binds to Fas receptor, it changes the shape of the death domain again, and that can bind to fat proteins. And the fat proteins become when the fat proteins and the death domain combine to each other, they make the another protein called disc, and this produces procaspase 8, and like before, we were gonna have the apoptosis like this picture here. It's like this before one.